Thank you very much for the kind introduction, and I'd like to take this opportunity to thank the organizers for giving me this opportunity here to present the in vivo cancer tissue models. So I just want to add a bit more about my background, just a brief word, because I'm quite conscious that my background would be quite different to a lot of the audience here. So what I do on a day-to-day basis is basically take a material and try to identify all the sort of variables that can influence cell behavior, and trying to alter them and control them in a way to control cell behaviors when the cells are placed into the material. So this is so that we could sort of mimic the tissue environment of tissues in health and diseases. So just a brief outline of the presentation. I'll start by talking just quite simply about the models that are currently available, and go into some more details about the merits of in vitro tissues. And then I'll go into discussing the considerations of the tissue microenvironment and how that may affect cell behavior. And finally, I'll go on to collagen-based in vitro environments, which can be controlled in a variety of ways, and how this may be applied to cancer tissues. So just really briefly, the aim of tissue engineering is to try to mimic the natural tissue environment. And there are two main branches that, of the outcomes that we aim to do. The first one is to create tissue implants, and the second one is to create model tissues. They have very different priorities in terms of the designing of the material, the platform. In the implants, we aim to create a material that is first and foremost safe for human implantation, and that will be able to functionally replace the lost tissue. For the second part, in tissue modeling, the most important aspect is to create a reproducible and reliable model so that we could reliably predict the native tissue response when we want to study the cells. So there are various types of tissue models, as we've seen already in the symposium, but just briefly, one of the models that is commonly used is the in vivo model. This, for example, will include the PDX models for cancer research. And they mostly are used to study whole system effects and the environment that is outside the immediate tumor tissue. However, there are considerations due to the time, cost, and ethical considerations. And also, quite importantly, there is also a question of whether some tissue models, some animal models, will be relevant to the human pathology. The next step down is the ex vivo tissue models. They are, for example, tissue slices that you extract from the animal, and then basically you have the cells that are preserved in a native environment, so that you could study how all these native cues affect the cells that are within the matrix. The drawback is that it's quite complex, so it's quite difficult to identify which factors are affecting the cell behavior. And so there's another model that is more simple, where you can isolate components to control, isolate components and see which components are affecting the cells. The drawback, of course, there's the removal of the cells from their native environment. So it's quite important that you're able to create a scaffold, a platform that at least has the minimum amount of the cues that the cells recognize in order to encourage the cells to behave as they would in the native tissue. So there are two main approaches to producing a in vitro model. The first one is to basically essentially take the ex vivo construct and then remove all the cells and all the impurities so that you're left with a scaffold that is relatively similar to the natural tissue matrix. The second approach is to build from bottom up, which basically takes the natural building blocks, for example, proteins and chemical cues such as growth factors, and then introducing them one by one into the model so that you could study which of these components affects cell behavior that you're looking for. So in short, the in vitro tissue model is aimed at understanding the tissue at the cellular level, and the aim is to trying to predict cell response to a stimulus within the tissue context. This is quite useful when you're trying to develop and refine therapeutic interventions, such as studying drug safety and efficacy. However, the key to all of this is that you're able to access the in vitro assays so that you could study the cells in lifetime, as opposed to in vivo models where you have to wait until the endpoint and take the, for example, cancer mass out for study. So when this is translated into in vitro cancer tissue modeling, we first want to make sure that it reflects human cell biology that is within the tumor tissue itself and also the surrounding tissues, which plays a part in the cancer progression. For example, the vascularization and the stroma cells and immune cells. This is so that we could study cell migration, the crosstalk between cell types, the vascularization of the cancer mass, which is important for its growth, and also how the therapeutic interventions will be able to stop the progression. So in order to do all of these, it's first important to identify what the native tissue environment is and what sort of components that you need to build into this in vitro model to create the cancer tissue of your choice, which obviously will depend on the tissue type. So you have to first define the tumor microenvironment and then define the extent of complexity that you want in your tissue model. In terms of the tissue microenvironment, this will include the cancer cells, obviously, and also non-cancer cells that are around the environment. These are all known to influence the tumor initiation, proliferation, and migration to a certain extent. And so you need to recreate the tumor microenvironment and identify the physical and chemical cues that play a part in the cell biology. So one of the main physical cues in the in vitro models is what the cell senses when they're attached to the surroundings. So the traditional in vitro model is the 2D model, which means cells are just placed on a flat tissue culture plate. However, we know that cells behave very differently. For example, cancer cells lose their in vivo features when placed in 2D. And so you want to try to mimic the architecture that is around the cells, and you could do this by placing cells in a 3D matrix that they recognize and they can manipulate. Other considerations for the physical environment include mechanical properties, so the stiffness of the material and the sort of the sort of architecture and other proteins that you might find in the natural cell environment. So another important consideration is what your material is made of. So there are two main types of material classes. The first one is synthetic materials. They're favored because of their highly controllable construct physical properties. So it's quite easy to control the pore size, and it's quite easy to coat the material with proteins that the cells recognize to create a usable scaffold. The second class is the natural materials, which includes, for example, collagen, fibrinogen, silk, and many others that you find in the natural, in nature, basically. And they provide provide the cell matrix interactions that are essential to direct cell behavior. So just to go into a bit more about the collagen material itself. So it is the most abundant protein that is found in the human body, and it is the main constituent of the extracellular matrix. It provides structural support and, importantly, provide the cells with attachment sites and cell matrix interactions, which will enable the cells to attach, migrate, and direct their behaviors. Collagen is quite interesting as well to study because the cancer matrix, the tumor the cancer matrix around the tumor also contains collagen proteins, and this enables the remodeling of the matrix can be modeled by using a collagen scaffold. So the main advantage of using a collagen hydrogel is the speed in which you can generate a native tissue-like scaffold with interstitial cells. So essentially you could place cells into the scaffold as it is being formed, and there's no need for a lengthy and variable cell seeding stage. It's possible to customize the construct in terms of the shape, size, and the composition. So you could add different proteins such as laminin and fibronectin, which all interplays and it influences how the cells behave within the model. However, there is a limiting factor of the collagen hydrogel scaffolds, and that is because it contains a high percentage of water, more than 99%, it often lacks the mechanical properties that you often associate with a native tissue. However, the lab that I've been working in has come up with a way to try to improve this by developing a process called plastic compression, which is quite simply using a weight and absorptive plunger that removes the excess water from the hydrogel, so that you could increase the collagen density to about 10%, which is closer to what you might find within the natural tissue matrix. So in terms of how this might be used in the cancer tissue models, this is work that have been done by my colleague at UCL. And basically what it is a two-component hydrogel matrix. The first component is a tumoroid seeded with colorectal cancer cells. There are two cell types tested, the HT29 and the HCT116 cell lines. And within the tumoroid is then seeded nested into a wider matrix that will act as the stroma surrounding. And this can be controlled in terms of collagen density, because we have the toolkits now to do this. And so we test, in effect, this tests the effect of tumor migration, metastasis, within a stromal surrounding that is either high in collagen density or low in collagen density. What they found was that when you seed HT29 cells within the tumor matrix, the cells migrate into the stroma cells quite differently in different patterns. In the low density matrix, they migrate out as smaller clusters. However, when you increase the collagen density to 10%, the cells detach from their tumoroids core and then migrate outwards as a more solid mass. However, when you look at the different cell types, the HCT116 cells, they tend to migrate out not as clusters, but as cell sheets. And so you could model the cell behavior and study how they differ. Also, in this model, they've added laminin to see how that will affect the cell migration behavior. And they found that in HCT cells, the addition of laminin appears to increase the rate of the cells migrating out of the tumoroids. So this, we know that the stromal surroundings and the cell density, the extracellular matrix density affects tumor cell migration. So other things you could study is the effect of oxygen gradients and nutrient gradients. This can be easily incorporated into the scaffolds by altering cell densities, as high cell densities will obviously use up more oxygen and nutrients within scaffolds. And then other factors that can be studied are vascularization of the model, which is usually quite difficult to study in 2D. But then in 3D models, you could co-culture endothelial cells in the model, and they will generate a vascularized matrix in hypoxic conditions. So another interesting aspect is to co-culture cells. As already multicellular spheroids is already widely used in preclinical screening, and they know that the co-culture of cells will interplay on how the cells react to drugs. So here, another thing you could study is how the immune cells and stromal cells interact with each other. For example, immune cells can acquire distinctive characteristics. For example, the M2 subtypes of the macrophages, which has a role in tumor progression. But then when you add the fibroblasts into the model, you'll see that you may see that the fibroblasts will enhance this by encouraging the macrophages to differentiate into the M2 subtypes, which exacerbates the progression of the cancer. So in terms of controlling the extracellular matrix environment, as I mentioned before, previously the hydrogels is lacking in mechanical properties. So in order to generate a even more dense and stiff construct, it is usually necessarily to artificially cross-link the collagen scaffolds. However, this has a drawback, as it is usually cytotoxic to cells, and it immediately removes one of the main advantages of using the hydrogels, which is the ability to seed cells at time 0 as the hydrogel is being formed. So the next generation hydrogels that I've been working on is to increase the stiffness without damaging the interstitial cells. So to do this, basically I introduced a new starting material into the hydrogel. This is a naturally occurring collagen, which is found in most tissues, and it naturally contains a high proportion of cross-linked collagen. So normally, the reason why this promising material is not usually used in a hydrogel setting is that it usually collapses into fibrous tissues when you neutralize the solution. So in order to incorporate cells, you need to make sure the the collagen doesn't collapse and inhibit these cells from entering the collagen matrix. Quite simply, what we found is that when you put the polymeric collagen into the conventional gelling collagen, which is acid soluble, and then mix it together, this prevents the polymeric collagen from collapsing immediately and enables a window to incorporate cells into the scaffold. So with the polymeric collagen, we're able to create a stiffer gel, which is about 3 times stiffer than what is already a compressed gels, which contains 10% collagen. And then we also increase the collagen density within the scaffold. This can be controlled by altering the amount of polymeric collagen you incorporate into this blended gels. However, this gives you another tool to control the collagen density. And so basically, just to keep a really long story short, this is just to show these cells can be incorporated into the cell surrounding the polymeric collagen containing gels. And this is an image at day 7 where you see the cells are still viable and able to be influenced by the cell surrounding. So in summary, collagen hydrogels is a promising platform for in vitro tissue engineering because you could incorporate cells into a hydrogel scaffold relatively quickly. They are highly accessible for assays and visualization, which is key to study the cells in real time. And it is a simple platform, but you can build them up into a sort of tissue-relevant models by incorporating a range of features. For example, you could incorporate growth factors and other physical cues that I've mentioned before. And I've presented some work on trying to solve the limitation of the collagen hydrogel, which is the lack of mechanical properties, so that we're able to have another tool to alter the cell surroundings environment. So in terms of future directions, this may be able to serve as a 3D in vitro platform to further our knowledge in cancer cell biology when you're able to incorporate and identify and incorporate the tissue-specific factors that makes the tumorous tissue as they are. And this will help in further studying of these drug delivery systems and cell response to therapeutic interventions and cancer progression. Thank you very much for your attention.